how a nerve cell gets excited, how it can depolarize the threshold, and how it can generate an action potential. Now we're picking up from there. Well, what happens? What's the point of that? Well, the point of a nerve cell generating an action potential is so that it can release its messenger that we're going to just call neurotransmitter for now, and we'll differentiate in more detail in a little bit. Because if you go back to chapter 5, we said the whole point of the body is to allow for intercellular communication, cell-to-cell communication. And the way that our nerve cells talk to our target cells is by way of neurotransmitter. So hopefully you're starting to see it weave together, come together and make a nice picture for us. But what makes it a bit difficult initially is that there's a whole bunch of events that lead up to the release of a nerve cell ever releasing its neurotransmitter. And it's been very well studied and very well characterized, hence the detail that we're going to enter into. We concluded class last time with a brief animation of what happens when a cell releases neurotransmitters. So we'll go ahead and label that on your guided note. Just to review some terminology, we're going to use the term presynaptic cell and postsynaptic cell. What's the reasoning behind that? At a synapse, a presynaptic cell is the one that releases the neurotransmitter. And the postsynaptic cell is just a very specific name for the what? The target that has the specific receptor. So we can label the presynaptic cell, and we can label the postsynaptic cell as well. So in this image, the presynaptic cell is the one on the top. The postsynaptic cell is the image on the bottom. Reminding you from our discussion on Thursday, this synapse may be at the target cell's dendrites, and we will call that a what?
Axodendritic.
Axodendritic synapse. It may be at the postsynaptic cell body, and we would call that
Axosomatic.
Axosomatic. Or maybe even at the hillock, we would call that what?
Axoaxonic.
Axoaxonic. Wherever this is, at a synapse, postsynaptic cell better have a receptor. How that receptor works takes us right back to chapter 5. The channel, G protein, right? Whatever the case may be. But before we can get to how it works, we've got to see the first release of the messenger. So the first thing is the presynaptic cell has to become excited. Again, whether that be chemical, mechanical, thermal, electrical, whatever, whether it's your ions, your photoreceptor, whatever, it's got to get excited. It's got to depolarize to its threshold and generate an action potential. So the presynaptic cell has to receive some stimulus, has to depolarize to its threshold and generate an action potential. We studied last week that action potential conduction depends on if that cell is myelinated or not, and other factors as well. So all that background information. But at some point, the action potential is going to reach the what? The axon hillock. Threshold's already been met. Action potential is being conducted, and it gets conducted to the axon terminal. So we're at the axon terminal of the presynaptic cell. So we're changing the voltage across the cell. And we know that we're opening and closing different voltage-gated sodium channels, potassium channels, but now we're adding something new to the mix. What is new here at the end of the axon terminal? Voltage-gated calcium channels. Go ahead and label it as such, that it's voltage-gated. These are going to open only when we change the voltage to generate an action potential. We open our voltage-gated calcium channels at the axon terminal, the presynaptic cell, when an action potential is present. Calcium is flowing into the axon terminal. So it has a nice little concentration gradient. Calcium comes into the presynaptic cell. So yes, calcium's charged, but it's not really contributing to the voltage. You may recall that calcium is the second messenger vesicle with neurotransmitter is exocytosed. So that the neurotransmitter is released into the synapse. So that an influx of calcium allows for those vesicles to actually migrate to the membrane and exocytose their contents. Y'all with me so far? That's the exocytosis of the neurotransmitter. Now the neurotransmitter is in the synaptic plaque. It's no longer bound, right? It's free. Where it should do what? Most of it's probably going to bind to a receptor, its specific receptor, at the postsynaptic cell. Dendrite, cell body, wherever that is. So once the neurotransmitter is released, it can bind to the respective receptor. So maybe you want to make a note. What happens here depends on the structure of the receptor. So that causes the response within the postsynaptic cell. That depends if you're looking at if this is a nerve cell, either a muscle cell, whatever, right? Depending on what it is, depending on the receptor, it can dictate what that cell does. In response to this, that's just telling you about whatever neurotransmitter.
What was 4 and 5?
4 is just binding of neurotransmitter to the receptor, and then 5 is the response within the cell, the target cell. But as soon as that neurotransmitter is present, you need to get rid of it. Because once this postsynaptic cell is being stimulated, it can't receive a new message. So we need to excite it and then get the messenger away. Does that make sense? Let the neurotransmitter do its job and then be done. So there's a few things that may happen to a neurotransmitter once it's been released, after it's bound. One way that we can see the neurotransmitter dealt with is by identified here in this image point 6 as an enzyme. Could catabolize it. Break it down as you might want to break it up, chop it up. We just change the what? We just change the chop it, I change the structure of it, and it can no longer
Do its job.
function, right, to do its job. Very good. So one way to get rid of the neurotransmitter is by enzymes that can cleave the neurotransmitter, as shown here in point 6. Alternatively, the neurotransmitter may actually be taken back up into where? You can see in point 7. It may be taken up where? Back into the presynaptic cell. And once it's back in the presynaptic cell, it's certainly not in the cleft. Enzymes can break down the neurotransmitter quickly, so they're not overstimulating the postsynaptic cell. So they're there to remove, as soon as the enzyme does its job, we can get rid of it.
Were you just pointing to the exocytosis?
Yes. And then once that neurotransmitter is gone, the postsynaptic cell has done its response. Both the cells, if they're nerve cells, go back to what? Resting membrane potential. And if a new stimulus is present, we can repeat the process. Or not until there is a stimulus present.
So, the receptors enzyme either degrade the acid.
In this case, in this scenario, that's it. The question was, what else does the enzyme do? That's what it does. Catabolize neurotransmitters, so it's not overexpressed.
So it doesn't really affect the iron.
No. That's its job. Get rid of those neurotransmitters. Kinda. Yeah, the messages come, told the postsynaptic cell what to do, and then the messenger has to be taken away. Otherwise, the postsynaptic cell would be falsely overstimulated. If that neurotransmitter hung around, it might be just really overstimulating the postsynaptic cell longer than it needs to be. So let's just recap. First, the presynaptic cell has to receive a stimulus, get excited to cross its threshold and generate an action potential. Action potential reaches the, you call the axon terminal, synaptic knob, synaptic bulb, synaptic terminal, right? The end of it. Where there happens to be a whole lot of voltage-gated calcium channels. So that potential change opens up the voltage-gated channels, voltage-gated calcium channels, and calcium flows into the cell, down its
Gradient.
electrochemical gradient. Very good. We're not really worried about changing the polarity here. What are we worried about? The calcium. We want what? It acts as a messenger. So we want to do something to this neurotransmitter. What's the process called? Exocytosis. The calcium triggers exocytosis of our neurotransmitters. They're exocytosed into the synapse, so they can diffuse across and bind with receptors of the target cell, or you could say postsynaptic cell. The effect depends upon the postsynaptic cell's receptor. Is it a gate? Is it a second message? Whatever, right? It depends. Once the message has been received, the neurotransmitter has to be, it needs to be catabolized or taken back up, or maybe even it just diffuses out. There's not in a synapse, not exposed to the receptor. And your astrocytes can go around and at least they can pick up stuff that we want. So the 2 weeks we've been talking about, we finally got to the culmination here. The whole point is to release neurotransmitter by changing the electrical voltage inside of a nerve cell. All that electrical change leads to the exocytosis of our neurotransmitter, which then is now an extracellular messenger. Binds the receptor, causal effect to the target cell. Do y'all want to see the animation again from last week? Will it help? Just reminding you, in this animation, the action potentials will be highlighted in yellow. This image is good because it helps put into perspective what we're looking at. Nerve cells that are comprised or make up our brain, our spinal cord, our cranial nerves, and our spinal nerves. All these cells have the ability to generate action potentials and release neurotransmitter. You can also see the little ganglion. In this presynaptic cell, we can see the microtubules, where our vesicles will be transported through. And then once the action potential reaches the terminal, we noted that in order to release those vesicles, we first have to see an influx of calcium. And those are highlighted in this next portion. We open up the voltage-gated calcium channels, calcium floods in. A whole series of reactions not shown, but the effect is those vesicles migrate, fuse, and purge the neurotransmitter out into the synaptic cleft. Now, the whole point of the neurotransmitter is to actually bind to receptors at the postsynaptic cell. And then what happens to the postsynaptic cell depends on the receptor. Does that help a little bit? This animation goes ahead and reviews what we studied last week and the effect of the postsynaptic cell. So let's just review that about an inhibitory versus an excitatory postsynaptic potential. If the synapse is inhibitory, which ion is it moving probably?
Potassium.
Potassium is probably flowing out, causing the cell to hyperpolarize. That's what we'll see first. And then conversely, at excitatory synapses, the neurotransmitter will allow sodium to move into the postsynaptic cell, causing what? If it's at rest, depolarization. Although this animation shows both scenarios, a synapse is either always going to be excitatory or it's always going to be inhibitory. You won't find a variable like maybe this animation might imply. Instead of showing two animations, just kind of stack them together. The receptors are built to do one job, not two different jobs. So in an inhibitory postsynaptic potential, we would see chemically gated potassium channels open up, a potassium would flow out of the cell. And the effect would be hyperpolarization in the postsynaptic cell. Does that make? In the reverse scenario, if this was an excitatory synapse, we would actually influx which ion? Sodium. And then if it's enough depolarization, the postsynaptic cell might cross its threshold and generate an action potential as implied in this yellow. Okay? The rest of this animation acknowledges what we just saw about the neurotransmitter. First thing is, this is the animation someone put together, so they're not all like in spaceship motion, like they're like going up to another ship. That's not going to happen like that. But it also shows some enzymatic activity, showing them actually being cytosolic. So we'll see some enzymes come in and cleave up the neurotransmitter. We can also see them being taken back up into the presynaptic cell. They may even just diffuse on out of the cell. But the big thing is they're not continuing to stimulate the postsynaptic cell. What questions do you have about these events at an axon terminal or even at the postsynaptic cell before we look at some specific types of neurotransmitters? Okay? Pretty confident I showed this image last week, but I think it's a cool image, so I'll show it again. This is a slide off the tip of an axon terminal where we can see highlighted in the blue and purples are vesicles which would have neurotransmitter in them. And then we can see the axon membrane, which is heightened color in the red, so she would be area. Alright, so I kind of alluded to this a moment ago, but what we can't forget is all the information we learned in Chapter 5 in this intercellular communication. We know that the postsynaptic cell is going to have a receptor, but we also studied that the receptors just may be chemically gated ion channels. So if we see a chemically gated ion channel, we're going to open a gate for a specific ion, right? And that would change the what of the postsynaptic cell?
Voltage.
The voltage, right? So if sodium floods in, the cell would, you said, depolarize. Potassium flows out, the cell would hyperpolarize, whatever the case may be. So we learned that our neurotransmitters at the postsynaptic cell just basically change the voltage by directly opening or closing ions. I'm going to say, you remember that? You're going to, yes, I got that from chapter what? Chapter 5. But we learned that the postsynaptic cell response may be a bit more complex when we pulled in your favorite second messenger systems. Like G protein, that, and that. And so our postsynaptic cell may have a G protein associated with it, like we learned in chapter 5, which may open a lot of ion channels or send them over to a whole other group. It may not be as simple as we just saw in the previous animation, open, closed, directed ion. So again, thinking, what am I talking about? We covered it in chapter 5, so make a reference to go back to chapter 5 about target cell response to what type of messengers, what type of solubility? What type of solubility is our messenger? Hydrophilic. Okay, so look at your hydrophilic messenger responses. So I'll just reiterate the role of the ion channels in the second messenger system. What I want to focus now is, instead of just saying neurotransmitter, we're going to get a little bit more specific on some different types of neurotransmitters that we'll see used in the body. I tried to identify this in the video a moment ago, is that the same neurotransmitter is always released at a synapse. If a cell is designed to release acetylcholine, it will always release acetylcholine. If it's designed to release dopamine, it's going to always release dopamine. Does that make sense? And so if the same neurotransmitter is always released from the presynaptic cell, the postsynaptic cell better have always the same receptors. So at a synapse, if a presynaptic cell releases acetylcholine, postsynaptic cell better have an acetylcholine receptor. It's always going to be consistent like that. And because the receptor is the same, the structure is the same, what else is the same?
Function.
The function and the effect. So at a synapse, the postsynaptic cell will always do something. It will always depolarize, or it will always hyperpolarize. Or if it's a muscle, it will always contract. Or if it's a different muscle, it may always relax. It just depends on the target cell and the receptor. But it's not going to, let's say this is the nervous system talking to your skeletal muscles. When your nervous system releases neurotransmitter, skeletal muscle might contract now and then take a break later, right? It's going to always do the same job. Contract, contract, contract. Another synapse somewhere else, they have a completely opposite. Looking at different receptors. So I'm just going to repeat this. The receptor-neurotransmitter relationship is consistent at any particular what? Synapse. Between synapses, right? Different synapse, maybe a different neurotransmitter, different receptor, different cell type. Is that acceptable? We actually just kind of hinted at what happens when a target cell receives neurotransmitter. If we're looking at a nerve-to-nerve type of response, they said the synapses can either be excitatory or inhibitory. Excitatory synapses are due to the influx of sodium. You guys like correctly identified. What type of polarity change happens here? Depolarization. If an excitatory synapse is present, the influx of sodium causes the postsynaptic cell to depolarize, get closer to its threshold. Whether or not threshold is met depends on the amount of sodium influx. Whether or not we depolarize to threshold. Get closer, but we may or may not get to it. Conversely, some synapses are considered to be inhibitory. You guys are correct that we may open potassium channels, and potassium will flow out, causing hyperpolarization. Not addressed so far, but I think I may have mentioned it, I don't know. But an alternative is we may open up fluoride ion channels. Fluoride has which charge? Negatively charged. So fluoride should move into the cell if I want it to hyperpolarize. And so if I open up chloride ion channels, it can also cause the postsynaptic cell to hyperpolarize, get further away from threshold. So a synapse, if it's considered to be excitatory, that means a postsynaptic cell depolarizes. If a synapse is inhibitory, that means a postsynaptic cell will hyperpolarize whenever the stimulus is present. And the relationship between the neurotransmitter and the receptor is consistent at a given synapse. If we look at some variations of neurotransmitters, table 8.1 illustrates for you where they are built from. Some come from, one comes from choline, it's called acetylcholine. Another comes from amine groups, which you've heard of dopamine, epinephrine, norepinephrine, maybe serotonin, you've heard of it. These are amine groups. Actual amino acids can function as neurotransmitters, including glutamate, acetate. It may be things that you didn't think of. ATP, ADP can act as neurotransmitters. And other kind of non-typical, non-classic things that we think of doing something else have neurotransmitter activity inside of the brain. So we're not going to study all of them. We're going to pick a few and going to focus on for the rest of the semester. This is in table 8.1, but I also have some structural images for you in your guided notes. So I'm going to kind of show the orientation of this table, and then we'll look at detail. First thing is, am I going to have you draw the chemical structure? You're going to tell me no. Maybe you'll do that in biochemistry. I wouldn't even know.
Yeah, really.
But what I like about this image is it talks about the receptors. If you remember, we said our receptors just may be simply gated channels, right? Or they may be what? Just show the image. Our receptors may be channels, or they may activate a second messenger, which is summarized here in the 7 helix. So where you have 7 helix, just say parentheses, G protein receptor, which activates the second messenger. So our receptors, we know these things are hydrophilic, so our receptors could act as an ion channel, or they could activate a G protein. Does that make sense? That's summarized. And then the rest of the list are some common neurotransmitters. Where you see a big dashed line, that means that particular receptor, or excuse me, neurotransmitter doesn't have that class of receptors. So for example, dopamine only binds to receptors that what? Activate G protein. They don't have just a little channel of them. They've got to activate a second messenger. The reverse is true for glycine. It only opens up chloride ion channels. Norepinephrine activates a second messenger. But some some neurotransmitter, some receptors are channels and some are G proteins. And so we've got some diversity in there. I love that, right? Multiple variations. Alright, so let's look at acetylcholine. Put a star on acetylcholine and put a star on norepinephrine. We're going to use these the most for the rest of the semester. I want you to become the most familiar with these guys. Acetylcholine first on the list, and norepinephrine second from the end. Acetylcholine can bind to receptors that act as channels, or it can bind to receptors that activate second messenger, the G protein. The name for the receptors for acetylcholine that act as channels is here in parentheses. What can we call these? Nicotinic. Because they bind to something that looks familiar, right? Nicotine. Right? They got someone's chemical right in our lab. Nicotine, that right? So they're called nicotinic receptors. Go ahead and just commit that one to the old memory. We're going to use it a lot. That are nicotinic remove the cause, the postsynaptic cell. But they are considered to be excitatory, so really it moves more sodium into the target cell. It moves more sodium in than it moves potassium out because we're going to say it's excitatory. We said something's excitatory, go back to your notes. We said it's going to let more sodium in. So our nicotinic cholinergic receptors allow sodium into the postsynaptic cell, causing the cell to depolarize. There are other types of cholinergic receptors. We call them muscarinic. These are the ones that activate a second messenger. And you notice we don't see the word excitatory or inhibitory on here. Notice the absence of any indication. So that means in some cells it may be what? Excitatory. In some other cell, acetylcholine may cause a suppression, an inhibition at the target cell. So it goes back to what we hopefully studied in chapter 5, that these G proteins can cause a variation of responses. So we can limit our receptors, but limit our receptors and open up our responses. These images are simplified for you in your textbook. And I've clearly omitted the page, the figure number, I'm sorry. Page number? 811, thank you. Figure 811. This is figure 811. The receptors that bind to acetylcholine that act as ion channels are called what? Nicotinic. But I want to turn your attention to this second word, cholinergic. Cholinergic. I take off the end part. What do I see? Choline. If I look at the name of this is acetylcholine. If I just wanted to be generic, I don't want to say nicotinic, muscarinic, I could just say cholinergic. Cholinergic has to do with acetylcholine. I could say a nerve cell releases acetylcholine, I could say it's a cholinergic nerve. If I said a receptor binds acetylcholine, I would call it a cholinergic receptor. Cholinergic is nothing to do with acetylcholine. Cholinergic just says acetylcholine, but it doesn't give us the function of the receptor. So if I want to know the function of the receptor, I've got to say nicotinic or muscarinic. If I say nicotinic cholinergic receptor, I'm talking about something that acts like as a channel, where it's going to let more sodium into the cell, causing the cell to depolarize. Nicotinic cholinergic receptors are always what at the target? Always excitatory. Very good. The effect is always excitatory of the postsynaptic cell when a nicotinic cholinergic receptor is activated. So you're stressing a little bit, but this is actually the easiest one. Okay? So it's just a gate. Remember we said our receptors be chemically gated ion channels. So a chemically gated ion channel example would be a nicotinic cholinergic receptor. We could also, though, our receptors could activate G proteins, and the receptors that receive acetylcholine and activate G proteins are called what? Muscarinic. And as I tried to identify a moment ago, depends on the cell if we're going to have an excitatory or an inhibitory effect in response to acetylcholine. The different kinases and everything activated depends on the cell type. Smooth muscle cells, whatever. The heart. So acetylcholine binds to two classes of receptors that are called nicotinic or what? Muscarinic. The nicotinic are just simply gated channels permeable to sodium. The muscarinic channels, what? They're associated with the G proteins. They activate the second messenger for the response. It could be excitatory in some cells. It could be inhibitory in others. It may not.
I mean more.
Or sodium. It's definitely not quantitative, but and your book may quantitate it. I'm not sure about it, but it's definitely got more sodium in than potassium out. Causing depolarization in the excitatory mode. So acetylcholine, we're going to see a whole heck of a lot, right, for the rest of the semester. So we'll keep using nicotinic receptors. All right. Another important neurotransmitter. Oh, excuse me. Let's add one more note about acetylcholine. I summarized that, or I could have said, we're going to see it all semester, because not only is it in the central nervous system, it's a neurotransmitter of the what? Peripheral nervous system. Hence why we're going to see it for the rest of the semester. Yes, it's used in the central nervous system, but we'll apply it for the rest of the body in the peripheral nervous system. There's tons. The question was, I thought we only had 4 neurotransmitters. And you could probably still add to that list. I'm sorry? You may have been just taught 4 because those are like usually focused on acetylcholine, epinephrine, norepinephrine, maybe dopamine and serotonin, but there's way more than that, including things that I mentioned that you wouldn't think is a neurotransmitter, like ATP is a good one. Common and most well known. The question is, why are we picking these of all the ones? We're picking these because these are the most common and widely used and characterized as well. So acetylcholine is a very important neurotransmitter for the peripheral nervous system. And as we go next in line, there's another neurotransmitter called dopamine. And what is its receptors built out of?
G protein.
A G protein. And it doesn't have a tricky name. It's called a dopamine receptor. So dopamine binds to dopamine receptors. We can handle that one. All right. So dopamine binds to dopamine receptors. I'm going to go back to that. Yeah, if you turn to the page 4 of your guided notes, there's a little bit of a note about dopamine. Dopamine is a neurotransmitter in our... Where do you hear about dopamine?
Central nervous
In
system.
the brain. So this is a neurotransmitter in the central nervous system. Dopamine is a neurotransmitter of the central nervous system. It binds to dopamine receptors, which are a G protein, activated with the G protein. Another highlight that we can talk about on this list includes norepinephrine, and I have you guys put an asterisk on this. Norepinephrine, its receptors are G proteins. And check out the name on this one. Everybody loves this one. What's the name of the receptors? Adrenergic. It looks like adrenaline. Another name for norepinephrine, noradrenaline, noradrenaline. So adrenergic, if I say adrenergic, I'm talking about what? You could say adrenaline, we're going to use norepinephrine, and there's another one called epinephrine. Or you could say, we're going to call it adrenaline noradrenaline. So if I say adrenergic, I'm talking about norepinephrine or even epinephrine. So a nerve could be adrenergic. That means that nerve releases epinephrine or norepinephrine. Or if I said the postsynaptic cell has adrenergic receptors, that means it receives norepinephrine and epinephrine. So let's go ahead and say that one. All together we're going to say adrenergic. Adrenergic. And it is a very important neurotransmitter also for the peripheral nervous system. That's why I had to put a star by it. So the neurotransmitters we're going to see in the peripheral nervous system is acetylcholine, norepinephrine, DEGRADATION, very similar structure, epinephrine. They are, yes, in CNS, but we're going to study them also in the peripheral nervous system. So they're in both. So adrenergic is talking about norepinephrine and epinephrine. Cholinergic, binding acetylcholine. Serotonin receptor binds serotonin. Another one that's not too tricky is NEUROTRANSMITTER. Before I move on, I want to see if you can see the variation. Some receptors combine to receptors that are neurotransmitters combine to receptors that are channel, and other receptors may act as second messengers decoding activated structures.
It doesn't really go with this. I guess it could, but is melatonin a neurotransmitter or is it something else?
Melatonin. It does
it's
come.
What helps you go to sleep.
Technically, it does go into the circulatory system, so technically, the pineal gland is called a gland, so I'm going to guess it is, I don't remember the histology of it, but I'm pretty sure it's endocrine in nature, and that's why we study in the endocrine system. So technically, it would be a hormone.
Okay.
The reason I was kind of hem-hawing is because there's some connection between the pineal gland and the optic nerve, and so there's probably some neural activity that I don't know about.
Maybe it's like a neurohormone?
It could be it may be classified as a neurohormone, I don't know, but historically it's categorized as a hormone.
Right. I was just reading.
Good question. All right. At the end of your Chapter 8 guided notes, we have some of these neurotransmitters listed. And they fall into the category of monoamine. Serotonin originates from tryptophan. You've heard of that? I guess we're about at Thanksgiving time. Amino acids, so there's a lot of protein. Tryptophan. Yes. As far as I know, I can't think of a cell type that would release serotonin. So serotonin is strictly a central nervous system neurotransmitter. Epinephrine, norepinephrine, they originate from tyrosine. And as we identified, these are also not only central but also peripheral nervous system neurotransmitters. Dopamine is, well, there's always something, right? Dopamine, when you call it dopamine, central nervous system, but there's a tie between it and oxytocin, which goes into the blood, so it depends on where you're studying it. But if you use the word dopamine, central nervous system.
Is epinephrine and norepinephrine released from the adrenal glands, or?
The adrenal medulla. And that goes right into chapter 11, so thank you. So, epinephrine, norepinephrine, dopamine, they're categorically called catecholamines. So later in the semester I talk about catecholamines. I may be just trying to say, instead of saying epinephrine and norepinephrine, I might just say catecholamines. Okay. Peripheral nervous system neurotransmitters, excitatory neurotransmitters. All of our monoamine neurotransmitters are often taken back up into the presynaptic terminal. So they're called uptake. We saw them in the image. They're often taken back up. Some of them can be catabolized by an enzyme called monoamine oxidase. An enzyme that oxidizes our
Neurotransmitters.
monoamine neurotransmitters. You've probably heard of this. MAOI, ever heard of those? The ads on TV. Someone's sad face, balloon broken. And they want you to buy some MAOI, monoamine oxidase inhibitors. Inhibit
Monoamine oxidation.
monoamine oxidase activity, which prevents the breakdown of serotonin and dopamine, which you've heard of, having to do with what?
Depression.
They could be involved in depression, but your overall emotional balance, right?
Right.
And so if you are categorically depressed, maybe your dopamine, serotonin levels aren't.
Serotonin is the one that, like, dopamine's the feel-good one, and then serotonin's the sad one? Are they both?
They're in balance.
Okay.
I don't know if there's a... Dopamine does get more of the feel-good, love, kind of happy category. I don't know about serotonin being in a bad way. But they have to be in proportion to one another to make your normal brain action. I don't know. The reason you want to inhibit a monoamine oxidase, the reason you want to inhibit an enzyme that normally breaks down a neurotransmitter, this would be pharmaceutically, right? A doctor would prescribe it. So if for some reason, whatever is going on, your neurotransmitter levels are low, if they're already low, below set point, and you start feeling sad or whatever, the gambling addiction is all awry, right? You can inhibit the breakdown of those neurotransmitters, and they last longer, so they would stay in the synapse longer, to allow them, hopefully, the brain function back to set what you want to do. That was just an example. All right. I think, does that conclude? No. We still have a little bit more. I wanted to address norepinephrine. We use the word adrenergic to describe those receptors that bind to epinephrine, norepinephrine. There's two families of them, alpha and beta. I like to say this. Alpha adrenergic, beta adrenergic. They're just kind of fun to say. There's two families of them, and they each have, there's variants of them. So if I use the word family, does that make sense? Within the adrenergic family, there's two subfamilies, the alpha beta, the alpha and the beta. And each of those have. Just like if I were to go to my family reunion, I have the Smith. Let's say, give me a name.
Smith and Jones.
Let's make up a name. Let's get real creative. Let's go with May. Okay? In the May family reunion, the Wilsons show up, but they're still in the May family, right? And then who else shows up? The Smiths, okay? So in the May family, we have the Wilsons and the Smiths. They're all binding norepinephrine, right? Within the Smith family, maybe there's 3 kids. In the Wilson family, there's 2 kids. Same thing in the alpha and the beta adrenergic receptors. There's variants of the adrenergic receptors.
So the alpha is more dominant and the beta is more passive, right?
You can categorically, I wouldn't use the word dominant and passive. I would use this, excitatory and inhibitory. Generally, the alpha adrenergic receptors cause an excitatory response in the postsynaptic cell. Generally cause the postsynaptic cell to depolarize. So notice the word general, not always. And generally, the beta adrenergic receptors cause an inhibition. The most notable famous exception is when norepinephrine binds to our beta adrenergic receptors at the SA node. If it was always inhibitory, the heart would do what in the presence of norepinephrine? Slow down, right? But we know that's not true. What does epinephrine do to your heart?
Speeds it up.
So I would modify your terminology. Alpha adrenergic receptors are usually excitatory. Beta adrenergic receptors usually cause an inhibitory response. Usually, with one famous exception. Beta adrenergic receptors in the SA node cause the heart to beat faster. It's a stimulatory and excitatory for the heart. You've studied this if you've been in a hospital. They're like, give me the beta blockers. Anybody done that? Because they don't want to say, give me the beta adrenergic blockers. You're crazy. You do something. Right? So they say beta blockers. It's easier to say beta than beta adrenergic receptors, right? What they mean is, if someone's in cardiac arrest, you can give a beta blocker, block the beta adrenergic receptor from binding norepinephrine, and so if we block the excitatory response, maybe the heart rate will go down and we'll get out of cardiac arrest. Does that make some sense? Sure. All right. Adrenergic binds
Norepinephrine and epinephrine.
norepinephrine, epinephrine. Cholinergic binds acetylcholine. Nicotinic. Cholinergic. It binds acetylcholine. The effect is always
Stimulatory.
excitatory. Stimulatory causes depolarization because it's just a little channel. All right. That is going to conclude our Chapter 8 discussion.